Hello everyone, and thank you for joining us, and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Tuesday, June 25th, 2013. My name is Brian Brewer, and I'm Director of Marketing and Communications for the Cancer Research Institute. The Cancer Research Institute is a nonprofit organization established in 1953 to support scientific research aimed at harnessing our immune system's power to fight cancer. Cancer immunotherapy is producing some of today's most promising new treatments, as you may have read in several prominent news reports over the past month. In light of these successes, the Cancer Research Institute has declared June Cancer Immunotherapy Awareness Month. We present this webinar series, 5 in total throughout the month of June, as a way to introduce you to some of the world's top scientists who are carrying out the important research that is leading to these cures. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how this new treatment modality is overturning how we think of cancer and how to treat it. After a 15 to 20-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your question at any time throughout the webinar by typing in the Q&A box on the lower right part of your screen. Again, there's a Q&A box on the lower right part of your screen. Please submit your questions throughout the webinar. I will do my best to get to everyone's questions. So before I introduce today's speaker, I'd like first to thank our sponsor, Dendreon, whose generous support has made this webinar series possible. Thank you, Dendreon. Now it's my pleasure to welcome today's speaker, Dr. Philip D. Greenberg. Dr. Greenberg is Professor of Medicine, Oncology, and Immunology at the Fred Hutchinson Cancer Research Center at the University of Washington in Seattle, Washington. His laboratory is involved in studies elucidating the immunobiology of host T-cell responses to infectious viruses and transformed cells. Dr. Greenberg's work has led directly to new immune-based cancer treatments that are contributing to today's quantum leap in cancer medicine. In 2011, Cancer Research Institute bestowed its highest scientific honor on Dr. Greenberg, the William B. Coley Award for Distinguished Research in Tumor Immunology, for his many contributions to this new approach to cancer therapy. He is a member of the CRI Scientific Advisory Council and a member of the CRI Ludwig Cancer Vaccine Collaborative, a global network of clinical trial sites with expertise in cancer immunotherapy. Dr. Greenberg, thank you for joining us today. We're delighted you could be here with us to give us your first-hand report from the front lines of cancer discovery.
Certainly, my pleasure. Thank you very much, Brian. I thought I should probably first just sort of introduce myself and explain how I got to where I am. At the time I was going to medical school and when I started training as a physician, the area of immunology was really exploding in terms of us being able to understand how the immune system works and how it prevents the development of disease. And because of that, I decided after I finished my medical training to actually take several years out and study precisely some basic biology about how the immune system works. And when I was done with that, I went back into my medical training and recognized that the area where it looked like this could really have an impact was cancer biology, cancer therapies. And that was actually, I hate to say well, let's say at least more than 3 decades ago. And at the time, it seemed like it was going to be an easy road, but it hasn't been an easy road. It's been a tough road to bring things out of the lab into the clinic. But we've done that now, and the opportunities now really look enormous. Originally, I did see many patients. I was trained as a medical oncologist. But in more recent years, the only patients I see are those really who are put onto clinical trials that emanate from our laboratory. And for the most part, even those patients are really monitored by people in my laboratory who are directly involved with treating them. So with that, I will proceed to the story I'm going to tell you today, which is really about cancer therapies and finding ways to selectively target and kill cancer cells. And this first slide is a picture of lung cancer actually growing. It's a 3-dimensional picture of lung cancer actually growing within the lung. You can see the air pockets, the empty spaces, and the cancer cells growing. And one of the problems is trying to get therapies that can specifically target cancers. And there's been a lot of work trying to do that because the original cancer therapies are kind of like using sledgehammers. You can hit the cancer, but you destroy a lot of normal tissue at the same time. And so people have tried to develop more targeted therapies so that you can actually hit the cancer and try to avoid it. And the idea would be something like that, where you could actually very selectively hit the cancer. And there's been an enormous effort both in drug development and in immune cancer therapy development, to try to actually get things that can selectively hit cancer cells and get rid of many of the toxicities that people recognize they're often subjected to in the context of cancer therapies. So one of these strategies has been to develop what are now called designer drugs, which are rationally designed, which means rather than just screening compounds, people have actually synthesized compounds that very specifically target something that's relevant to the tumor. So, for example, as we've been able to learn in the last, probably 1 to 2 decades, what are the selective abnormalities found in cancer cells that are responsible for the tumor, the idea is to develop drugs that can actually very specifically target those abnormalities. And two of the examples are drugs that are out there right now for treating CML, the drug imatinib, which targets the translocation of 2 genes, which is responsible for causing CML. It's very effective. There's another drug, Immunotherapy, which has been used in melanoma patients, which targets a mutation which about 40% of patients with melanoma have. It has also, again, been very effective. Both are very effective at inducing responses. But unfortunately, the problem is that cancer cells are actually hijacking these pathways. These particular pathways I talked about are used by normal cells, so the cancer cell is not creating them. And thus, when you try to block these pathways or disrupt them with drugs, there is still toxicity to normal tissues that are using those pathways. And in addition, it's turned out that the tumor cells are capable of developing strategies to bypass the inhibition that occurs with these drugs. And as a result, none of these drugs, despite their dramatic responses, have actually been curative. And so patients are ultimately relapsing after being treated with these drugs. So in summary, these designer drugs the approach has been very novel. It's really been bringing science from the laboratory to the clinic. It's been very exciting. They're very beneficial. But these drugs still do lack the fine specificity that's needed, and very few drugs actually find the true Achilles heel of the tumor so that the tumor actually goes away and doesn't come back. And consequently, this is where we believe immunotherapy can really step in and do a more complete job. So the seed for cancer immunology really began a long time ago with the idea that the immune system surveys the body to prevent the development of cancer. This was actually proposed incredibly in 1909 by Paul Ehrlich, who you may know won a Nobel Prize for his work developing a drug to treat syphilis. But he recognized that if the immune system was there to get rid of infections, it's probably there to get rid of tumors also. It was an interesting hypothesis. Of course, there was virtually nothing known about the immune system then, and there was no evidence that it was true. In 1957, really, nothing was really done for more than half a century, and then finally, Lou Thomas proposed in 1957 that the primary function of the cellular immune system, the T-cell part of the immune system, which had just been discovered in the 50s, that the immune system was divided into antibody-producing cells and T cells, which mediate cellular immunity, that those were there to protect us from cancers. And that was done basically based on the fact that if you transplanted a cancer onto a mouse, you could show that T cells were responsible for ejecting them. Now, there were lots of reasons why that was occurring in those experimental systems, and although Thomas was right, it is not the primary function of the immune system. The immune system really did evolve to protect us from infections, and that's one of the reasons why the immune system doesn't naturally help us ultimately get rid of many cancers. McFarland and Burnett in the same year agreed with Thomas, suggested it wasn't inconceivable that this was what the cellular immune system was doing, and that cancers ought to be invoking a response. Burnett was another person who won the Nobel Prize for his understanding of how the immune system recognized self and non-self. And so his idea that this was important for cancer became widely acclaimed. And in 1964, he actually, by that time when we started recognizing that cancers had genetic changes, he proposed that this was exactly what the immune system was seeing. these were all very good theories. They were logical. They were based on available evidence, but there was actually no experimental data that supported immunosurveillance. And that really didn't occur until the late 1990s when Bob Schreiber's lab at Washington University really proved that this does actually occur. So what is immunotherapy based on? Well, the premise, of course, is that tumors express antigens that the immune system can target. And there's been incredible enthusiasm and optimism for using the immune system. This has really increased with each decade. And I'm just going to highlight those findings that have really impacted our understanding for cellular immunity. So the first is in the 1960s, as I said, when Burnett and Thomas proposed that T-cell immunity evolved to protect us from cancers. In the 1970s was really the first beginning of evidence for that. Patients receiving bone marrow transplants, which were developed here at the center where I've trained, the Fractions and Cancer Research Center by Don Thomas, who won a Nobel Prize for developing bone marrow transplantation, which is now being used to treat has been used to treat now more than 1 million patients. And what was shown is that the reason why transplants work so well was because donor T cells were found to mediate anti-leukemic activity. In the 1980s, the transfer of T cells was shown to be able to react with tumor cells and actually mediate tumor regression. These were studies done at the NCI by Steve Rosenberg's lab. In the 1990s, patients who relapsed after bone marrow transplants, you could show that infusions of T cells from the donor. Let me try to turn this up. I'm afraid I'm getting some noise here. No, so much for that. As the only therapy that could induce complete remissions. In the 2000s, it was shown that vaccination of cancer patients with antigens expressed by tumors were found to induce tumor regressions. And then finally, in 2010, several cancer drugs were shown actually to work and were approved by the FDA as standard therapies. Provenge, a vaccine against prostate cancer, and ipilimumab, a drug developed by Jim Allison, who is the scientific director of the Cancer Research Institute now, has been shown to actually induce responses that can work in patients. So the idea now is to use immunotherapy to improve the selectivity of cancer therapies more than we could do, for example, with targeted drug therapies. So again, what we're trying to do here is harness a biologic strategy that evolved over millions of years to very efficiently protect the host from foreign invaders like bacteria and viruses. And what we want to do is harness that to target cancers. Now, most immunotherapies try to enhance the activity of one of the two major arms of the immune system, which is either the humoral immune response, which is largely represented by antibodies, or the cellular response, which is represented by T cells. But this is still a problem because even for the immune system, the tumor is still not sufficiently different. It's still a cell that evolved from a normal cell. And it's not as foreign, obviously, as a completely foreign pathogen, which shows up with its entire new genome. And so the natural responses that can be elicited are rarely sufficient or strong enough to provide benefit against established cancers. So largely, we and others have explored ways to provide or engineer better immune responses than the patient can actually make themselves, and to make these the immune responses that patients can use. So first, I'm briefly going to talk about antibody cancer therapies. What I've shown here on the top is this is an antibody molecule that binds, and this is a three-dimensional picture of it. These are where the antibody binds. It has two binding sites. They both bind exactly the same part of the same molecule. And by binding two parts, you can actually induce killing much more effectively. Strong antibody to human responses can be generated in mice. Humans don't generate very strong responses, but strong responses can be generated in mice because they see these human cancer proteins as completely foreign. And these antibodies that you make in mice can be humanized so that they look like a human antibody rather than a mouse antibody. So the genetic technology for changing this is readily available now. And so you can isolate these antibody-producing cells. You can clone this gene from the antibody, and now with synthetic biology, you can actually now make unlimited amounts of this antibody and produce it as a drug. And patients can then be treated by administering these antibodies, which are immunologic reagents, as drugs, and they can target the tumor, and you can provide these antibodies in amounts that could never be produced naturally in the individual. So this is just some example of some antibody therapies that are now currently approved for therapy. Rituximab in lymphoma, Herceptin in breast cancer, cetuximab in colon cancer and head and neck cancers. The problem with therapy with antibodies is that although they provide therapeutic benefits, they've, again, rarely been curative. And they also require, they don't generally kill the tumor by itself. They actually require recruiting and engaging an immune effector cell that can deliver what's the critical lethal hit. There are other places where antibodies can be enormously effective, and this is particularly in the prevention of cancers, and particularly those that are caused by cancer-causing viruses. Now, this obviously makes sense because we know that we can vaccinate against other kinds of viruses that cause infections. Most of the childhood vaccines are exactly that. And so the most dramatic successes have really been seen in preventing HPV-associated cancers, the human papillomavirus. These are responsible for 90% of cervical cancers, and it's been shown that this is virtually 100% effective against development of cancer if given before sexual encounter that actually induces HPV infection. So it's very important this be given to prepubescent girls before they have exposure to HPV. And unfortunately, we have not, the U.S. has actually been quite slow in actually getting this out effectively to the U.S. population, much less effective than other nations, which is rather disappointing. So we are, of course, focusing not on the antibody response, but on T-cell responses. And the response we're working on is CD8 T cells, and that's really how that's really a cell that's nickname is called a killer cell. And I'm going to show you a video in a second of how that cell got its name. This is the tumor cell, the very large cell. The small cell on the bottom is the CD8 killer cell. This kind of looks like a David and Goliath event where you have this very large, aggressive tumor and this sort of mild-mannered killer T cell. But that's not exactly what happens. And so can you play the movie, Brian? So this is going to be a movie then you're going to see of, this is the cell, the T cell now on the bottom, and well, very fast, but okay. But you can see what basic, there it is now. And you can see it essentially destroys the integrity of the cell. The tumor cell implodes. The cell actually continues and destroys the nucleus. And I think, and we can go back to the slides now, Brian. What's impressive, I think, is that this same T cell can do this over and over again. So it can kill many cancer cells, not just one. So this very small T cell can be quite powerful. Thanks very much. So what has to be addressed to make T cells work? Well, you need a good antigen. The tumor has to be expressed preferentially in the tumor, not in normal tissues. It's best if this antigen is not dispensable to the tumor, so the tumor can't escape it by losing that antigen. And you have to be able to elicit a T cell response. So that you have to find a way to make this antigen immunogenic. You need a potent T cell response. The T cell has to be of what we call high avidity. It has to see this antigen and bind very tightly, not loosely. It has to be of high magnitude in order to eradicate an established tumor. And the T cell has to be able to sustain its function. It needs to overcome the inhibitory pathways that may be operative, and we'll talk very briefly about some of the strategies that can be used to actually make sure that occurs. So the first step is to identify and validate target antigens. And the molecular technologies for doing this now are actually quite remarkable. You can take out the messenger RNA, which encodes all the proteins in a tumor. You can take out the RNA from its counterpart, the normal cell. You can then label them with a fluorescein probe, and you can hybridize them together on a gene chip, and you look by fluorescent detection, and you can find those genes that are overexpressed in the tumor, not found in the normal cell, by this simple technique of which one binds better, and you can select the genes that are appropriate for targeting. And we've done this now in looking, and we've looked at leukemia as a prototype. Sebastian Oxenreiter, when he was a postdoc in my lab, did a lot of this work in collaboration with Ravi Mujedi and Irv Weissman at the University of Stanford. So the first thing we did was we looked at the gene expression in leukemia. WT1 was an antigen that appeared to be a very strong candidate based on its differential expression in leukemia compared to normal cells. This is just looking at the level of expression in most leukemia cells compared to normal cells. You can see it's expressed at enormously higher levels in most leukemias. About 90% of acute myelogenous leukemias. We wanted to ask whether it could actually recognize the stem cell, the cell that's really responsible for causing the leukemia. So we looked at gene expression in leukemic stem cells that were purified by Ravi Majetian with our Weissman. And if you look here, you can see on the bottom, this is the heat map. You can see that in red that the genes are overexpressed very dramatically in red in leukemia, not in the top part, in the normal counterpart. If you look if you look, it's expressed through all the differentiation pathways of myelogenous leukemia. So the question is, will it be safe to target? So we've looked through all the normal tissues where you can find it. It is found in the uterus and in the testes. It's also found at low levels in normal adult kidneys. But really, WT1's importance is both in malignant cells as well as in the development of the urogenital tract, but not so much in normal adult tissues. We've looked to see whether we can find other targets beyond WT1. And so again, in collaboration with Ravi Majetian of Weissman, we looked at what we could find in preferential leukemic stem cells. When we did that extensive analysis, looking at virtually the millions of genes that are expressed, 50 genes fell out of that analysis. And of those 50 genes, there were really only 9 that looked like they actually would be viable targets. And when we looked, and these are the 9 I'm showing you here that are expressed in leukemic stem cells. And when we actually went through those 9, there was really only one that looked very good in it besides WT1, and that's another gene called cyclin A1, or CCNA1 is what the gene is called. It is a very new potential leukemic target. We described this last year. It's a family of conserved proteins. They regulate, these genes normally regulate cell cycle progression. There's really 2 family members. One is CCNA2. This is found in every cell in the body. It regulates cell division. But CCNA1 or cyclin A1 is found only in male germ cells, and it's actually responsible for a different type of division, meiosis, which occurs in reproductive cells. If you knock down CCNA1 in acute leukemia, it reduces proliferation. That's a good sign. It tells you it's important for the leukemic cell. If you knock out the gene in mice so that it's not expressed at all in the mice are sterile. That makes sense because we know it's important for germ cell development and division. But the mice are otherwise perfectly normal, suggesting it has no other normal function. And if you overexpress CCNA1 in mice in normal bone marrow progenitor cells, these mice develop abnormal myelopoiesis and develop leukemia, again affirming that this is really an important gene in the development of leukemia. So once you have a gene like this, such as this or WT1, how do you find out if you can generate an immune response? So Matthias Wolfe, when he was a postdoc in my lab, developed a system in which we could take the gene, we could make a range of all the peptides that could be derived from that protein, and add it onto dendritic cells, the most potent stimulating cell in the immune system, and use it to stimulate naive T cells. And if you do that, you stimulate them, you re-stimulate with peptide, we isolate the reactive cells, which are CD137 positive, we expand them, and then we screen them for which of the peptides they recognize, and using that we can identify not only whether we get a response, but specifically to what part of the protein that response is generated to. And using that technique, we can basically detect whether or not any particular T cell, any particular protein can elicit a T cell response in any individual. Once we do that, we isolate T cell clones that recognize those proteins, and we ask, particularly in leukemia, whether or not those cells can actually recognize the leukemic stem cell. And so in collaboration with John Dick's lab in Toronto, we used a system that his lab developed for looking at the leukemic initiating cells. So if you look in the leukemia population, most of the cells are more mature. They are not the cells that are actually causing the leukemia. They're derived from the cells that cause the leukemia. So if you take the cells out of the blood or out of the bone marrow and put them in tissue culture, you can have cells which actually can form what look like leukemia clones, but even most of those aren't really the true progenitor cell. But if you put these into these skid mice, these immunodeficient mice, you can show that about one in every 2 × 10⁵ cells that you find in bone marrow or blood actually are these leukemia initiating cells. And so what we do is we take the leukemic cells, we incubate them with the clone we've generated that we think recognizes potential candidate. We then put this into irradiated mice, these irradiated skid mice. We inject the mixture of cells. We support these leukemic cells to grow in the mouse, and we look. And if we've mixed the leukemia with a control clone, we see it grows out quite well. When we mix it with a clone that actually recognizes the leukemic stem cell, we see complete elimination or elimination of virtually all the stem cells, telling us we actually have a T cell that can recognize a leukemic target. So I'm going to switch now to a clinical trial we've done in patients with leukemia in AML. The strategy we use, we generate these T cells in vitro, we clone and select them, we can genetically modify them and expand them in tissue culture, and then we infuse them into patient. And before we infuse them into the patient, we can do things to the patient that will enhance the ability of the cells to persist. And one of the tricks that was developed actually by Steve Rosenberg's lab at the NCI is to lymphodeplete the patient so that the patient's body thinks it needs to expand its lymphoid population back to normal. It's a natural response we have to restoring homeostasis in the body. And if you lymphodeplete and then give a large number of cells, those are the cells that are there in abundance, and they will expand. They'll take advantage of that. We infuse the cells. They can expand to quite large numbers. We can provide things after we've given the cells to enhance their function. And again, these are a lot of strategies that we could talk about later if people are interested in the details. So this is what we've been doing in WT1. We have a bone marrow donor. We do a leukapheresis. We isolate the CD8 positive and negative cells. The CD8 positive cells contain the potential cells that will generate the response. We take the CD8 negative cells. We isolate the dendritic cells. We pulse them with a WT1 peptide. We grow these. We use that to stimulate T cells. We grow it with a panel of cytokines. We then do cloning of those cells. We expand these cells in flasks to cryopreserve them. And in about 10 to 12 weeks, we can make more than 1 billion cells from a single cell that recognizes this WT1 expressed in their leukemia, and then we infuse that into the patient as therapy. We have a clinical trial that was started by Gunnar Ragnarsson in my lab and has been completed by Aude Chapuis. This is what the trial looked like. Patients came in for transplant, which is HCT, is a hematopoietic cell transplant. We generated the cells while the patients were undergoing the transplant, and then we monitored them for relapse and then gave the cells and the doses in the system that you can see to the right. Subsequently, after we showed it was safe, we've actually then started treating some patients that were at high risk of relapse, at high risk of relapse rather than just waiting for them to relapse. And I'll show you some data to that effect. So this is one patient we treated who had, as you can see, based on the red is the number of blasts, had relapsed, had a high number of leukemic cells circulating in the blood. We gave the T cells, and as you can see, the leukemic cells in red came down to 0. They became transiently undetectable. This was very encouraging. In the gray, you can see the patient recovered their normal white count, which had been suppressed by the leukemia. This was, again, very encouraging data. But unfortunately, the patient at that point developed a toxicity, or a toxicity that the patient had developed from transplant that actually progressed. And because of the rules that were required with the FDA, we were not allowed actually to give the patient additional infusions of T cells. Unfortunately, in this patient, the leukemia relapsed, and the patient went on, unfortunately, to expire of leukemia. So this is a real problem in treating late-stage advanced leukemias, which are very rapidly progressive, and most of these patients have had a lot of therapy, and this may actually preclude being able to complete therapy, and so it is going to be preferable to treat patients earlier, and I'll show you some results in a patient we treated earlier. This was a patient who was treated who had a molecular marker where we could detect that the patient's leukemia had relapsed, although you couldn't see the cells. We could do it by molecular technology. And so we gave the patient T cells at that point, and what you can see is that the patient's, let me see if I can find that. I can't find the pointer, unfortunately. The patient's leukemia came back, it dropped down to, as you can see at day 20, it dropped down. Oh, there it is. Okay, good. Thank you very much. It dropped down to 0 at that point when we gave the T cells. That was quite encouraging. We then gave another infusion. But then over the next 2 weeks, the leukemia, you can see, came back. But we then gave another infusion of T cells. And then the leukemia really disappeared and stayed disappeared. The T cells lasted quite a while. You can see here at 5 months after we gave the cells, These cells represented about 7% of the cells in the patient's peripheral blood. This then really is the strongest response in this patient's body. It represented almost 10% of the cells in the patient's body, lymphocytes in the patient's body. The patient did quite well. And this is a summary of the data then. We actually enrolled 37 patients on that trial. Because we were waiting for patients to relapse and the problems as I talked about treating them, we only got to treat 11 patients on those trials. 2 of the 7 patients who had detectable advanced disease exhibited reduction in leukemic burden, but unfortunately went on and progressed. There were 4 patients who had minimal residual disease or no disease. These patients had a poor prognosis that was very predictive, and I'm going to show you a summary of that data. This was the patient I just showed you, had minimal residual disease when we treated the patient. The prediction is that this patient would have a median survival of only 5.9 months. This patient would be predicted to be alive at 2 years at only about 3%. This patient did die, unfortunately, at 28 months of relapse, but was disease-free for more than 2 years, far longer than what anybody would have predicted from the available therapy. These are the other 3 patients we treated. As you can see, these patients would be predicted to have a 50% survival of 2.8, 5.9, 7.5 months. Very little chance of being alive at 2 years. All of these patients are still in continuous complete remission at more than 3 years now out. And so this looks very promising. Because we didn't actually have detectable leukemia to measure, we can't say that the T cells did this. But based on these percentages, I think the likelihood that these patients receive benefit and may hopefully be cured because of this therapy, we have very good reason to be optimistic. And we want to make this more generally effective and more easy to give to patients. And so what we want to do is make sure that we can give every patient a high avidity T cell, and we can generate these cells very rapidly. And we're doing this by essentially a second generation technology where we don't actually have to clone the cells as I showed you for each individual patient in that last trial, but one where we can create these cells very rapidly in tissue culture. So basically when we have, for example, a clone we show works very well, rather than have to reproduce that in every patient, we clone the T cell receptor genes, which are what give the T cell specificity. So now when another patient comes in whose tumor shares those molecules, we take this T cell receptor and we put it into this patient's T cell so that now the patient's T cell expresses this gene we cloned from a cell that works. We now have a clone that we know expresses a gene that makes it recognize the leukemia well. We've developed techniques to modify those genes to make them express well and behave better. We know that we can do things. We can select cells from the patient that will improve the way these cells work. And again, I'm not going to be able to go through that. Well, once we put these genes, we put these into we've actually hijacked the technology. We use a modified strategy of an HIV virus where the HIV part has been disabled, but we put the T cell receptor into the HIV virus and use it to infect the T cells, to introduce the genes, and we get high level expression. The vector we're using was actually developed by Rick Morgan in the Rosenberg lab at the NCI, and they've very kindly provided to us for these studies. And this is the kinds of collaborations across labs that really advance cancer therapies very rapidly. So this is an example of the relative avidity, the relative binding capacity. The higher up on this scale, the better it binds leukemia of the different clones we gave to patients. We then went ahead and screened more than 100 donors, literally thousands of clones, and found one that bound leukemia much better than any of the clones we had given to patients. And consequently, what Tom did is isolate the gene from that, showed that it binds, it's the one in red, it binds much tighter than any of the T cell receptors from any of the clones we gave. If we put it into T cells, the T cells we put it into recognize leukemia just as well as the clone from which T cell receptor was derived. This shows you it actually kills leukemia much better. It's this one in red, much better than any of the clones we gave to any of the patients in the previous trial. And so we've now started a trial. We're enrolling patients already. We've treated 3 patients with T cells transduced with this receptor. And we also have a trial pending in lung cancer that's being supported by the Stand Up to Cancer which is sponsored by the CRI and the American Association of Cancer Research, we're going to be treating cancer patients with this clone, collaboration between Stan Rendell's lab and myself. And we think we can actually make these T-cell receptors even better. That's because we understand what the T-cell receptor sees. It's the CDR3 region of the T-cell receptor that binds the peptide, and this is showing you which part of this T-cell receptor on top. This is the antigen where it's presented. It's the CDR3 region that binds. Here you can see again the CDR3 regions sitting on top of the receptor. This is the peptide in yellow. And again, this is one more picture of that. And what David Kranz, a collaborator of ours, developed was a technique where we can do mutagenesis through that region to find receptors that bind the antigen with much higher affinity. So we can express the receptor in a hybridoma. We can mutagenize it, saturation mutagenesis through that region, re-express all those TCRs, and then screen those receptors with tetramers to find ones that bind this antigen with higher affinity. And basically, we can reject the ones that don't bind with very high affinity. We can isolate the ones that bind with higher affinity, select them, and then express them. And this is an example of two that we found. This is the wild-type receptor. It binds very poorly if you don't add CD8 to it to help it bind. This is now what happens if actually you mutagenize it. So these are two mutants that bind with much higher avidity. And so we looked at those clones in mice. We put them into mice. They appear perfectly safe. That's the control receptor. These are the receptors that are higher affinity. These two. They don't localize in any of the tissues where we know WT1 is expressed in normal tissue. They don't get rid of normal hematopoietic stem cells, which express very low levels of WT1, so they appear safe and functional. And we asked, of course, if we can find these, if we can mutagenize these and they're safe, why aren't we finding these in normal patients? And it turns out that the answer, in order to ask that question, we watch what happens when these T cells develop in the thymus. And unfortunately, these slides didn't come out well, so I'm going to skip to the conclusion. What you can see is that what you can show actually is that these cells get deleted or modified in the thymus because the gene for this protein is expressed at very high levels in the thymus, so that the cells that come out from hematopoietic stem cells express low levels of the TCR. That's how they survive the deletion process in the thymus. But if you express them in the periphery, they actually respond much better. And if you look actually now in the thymus, what you can show is that's because this protein is expressed at very high levels in the thymus, despite the fact that it's not found in normal tissues at very high levels. And so what this is telling us is that the thymus is deleting a lot of receptors that would be very safe, that there is a margin of safety here where we can increase affinity, make cells see leukemia at a much better way, as well as many other tumors that express WT1, and we can actually use that for therapy. And that's really what our next generation strategy is going to be. So we are trying to overcome barriers to efficacy, designing the right type of T-cell response, modifying T-cells to localize the tumor sites that they'll respond better and resist some of that immunosuppression. I'm just going to summarize this data. We know there's very different kinds of T-cells, naive cells, central memory cells, effector memory cells. Stan Rendell in our group has developed technologies where we can isolate each of these subsets separately, and we can then put the T-cell receptor into any one of these subsets and use them for therapy. And some of our preclinical data in the mouse has shown that in different settings, different T-cell subsets are going to work better. In other settings, when tumor is either very extensive or minimal, different T-cell subsets are likely to work better. So we're developing the technologies now to actually put in the design, not only a designer T-cell receptor, but put it into the ideal T-cell that can mediate the effect that you need in that particular patient's disease state. We can also put in genes that make the cells overcome barriers. So we've talked about putting in receptors. Here we can put in genes that make the cells function better, improve their survival. We can put in genes that make the cells localize to particular tissues. For example, if the leukemia is in the bone marrow, we can put a receptor that makes T cells want to go to the bone marrow. And we could take advantage of many of since we know many of the genes that are used by tumors to suppress immune responses, initiate these inhibitory signals in T cells, we can actually block those pathways by putting genes into the T cells that make those genes not able to be activated, not to block those pathways, so that these cells can function even in the presence of those pathways. So right now, for example, antibodies to many of these pathways are being pursued clinically. These require global immunosuppression and can cause side effects, but we can actually put them into the specific T cell that's recognizing the leukemia, and we can actually therefore get rid of those toxicities and really make this a very effective targeted therapy. So I'm just going to end here. Cancer immunotherapy in 2013. There's enormous promise. Is this going to be the year that it's going to be fully realized? Well, not fully realized, but we've come in an enormous way, and there's more to come. And I think the promise is incredible, and much of it will be realized. Right now, this is a very exciting time in the field of immunotherapy. But I don't want to promise too much. I thought I would end actually with a quote from Milton Berle. For some of you out there, he may be too old. He sadly is not too old for me. But he made a very prescient statement, actually, even back in the 1950s, when he said that people who don't know why America is called the land of promise should be here during an election campaign. I think it will become very clear. But we don't think we're kind of promising the same things that occurred during an election campaign. But we know actually that it's going to take a while, but with the kind of support that institutes like the Cancer Research Institute is providing, it's going to become a real reality for patients in the very near future. Thank you.
Well, thank you very much, Dr. Greenberg, for that very excellent presentation. I, for one, was very encouraged by some of the clinical trial data that you shared with us, showing that some of those patients with advanced leukemia experienced remissions, and it does look like, although you say that the T cells themselves may not be, or you can't say definitively that that was the cause, the data certainly seem to support that conclusion. So my first question is, well, first let me remind everyone that you can take advantage of this opportunity to share your questions with an expert in the field just by typing them in the Q&A box that's at the bottom right-hand side of your screen. I'll be fielding those questions to Dr. Greenberg on your behalf. So, Dr. Greenberg, the first question that we got early on in the presentation, you said that cancer cells can hijack normal processes, normal parts of the immune system. Can you talk a little bit more about how cancer hijacks the body and how this potentially leads to complications in treatment?
Yeah, so cancer cells are cells essentially that have developed the capacity to divide what we call autonomously, that they develop a way that they can keep replicating themselves, even though normally there are checkpoints. So that even, for example, when you have an injury, you cut yourself, you grow back new skin, but the skin stops growing after it understands that it's actually done its job. Well, cancers lose those checkpoints. They don't stop dividing. They keep dividing, even though they should be getting cues that tell them to stop. So what cancer cells do is hijack pathways that tell them to keep dividing, keep proliferating, keep expanding, and spreading, things that normal cells stop. And so they are using normal pathways, they're just using them very abnormally. So some of this is by overexpressing them or by introducing mutations that make them persistently active so they don't respond to off signals. And these are problems that the cancer cell has that allows it to escape the normal checkpoints that stop it, but they're still normal, so that if you block that pathway, then, for example, it may be that you can't heal a wound because you no longer are able to generate, because that pathway has been blocked by a drug, or you've prevented cells from dividing that need to divide. And that's why, if you notice that with chemotherapy, most chemotherapies intrinsically target very rapidly dividing cells, and that's why people lose their hair. We all know that, as you can tell from me, hair grows rather quite robustly, as does, for example, your bowel replaces itself, produce new white cells. And so the cells that get targeted by chemotherapy, the toxicities usually represent cells that are forced to have to divide. So the tumor cells are hijacking this. They're using this in ways that allow it to actually keep going without understanding why it should stop. And so you have to find a way to make it stop.
Okay. You spoke a lot about leukemia, and you did reference that this is a technology that could be used for other types of cancers, which is one of the benefits that we hear so often about immunotherapy in general. How far is your approach now in treating patients with other types of cancers, and which ones are those?
So we have recently just got a grant now to treat treat lung cancer. We're just in the process of getting the protocol to do that approved. It'll be a collaboration between myself and Stan Riddell here, and supported in part by the Cancer Research Institute. That's because WT1 is actually, as we said, the tumor is hijacking that to help it divide and become malignant, but many tumors do. So it's overexpressed in the vast majority of lung cancers. It's overexpressed in the vast majority of patients with pancreatic cancer, ovarian cancer, and so we are actually looking at ways to target it in those diseases. We have some preclinical studies in pancreatic cancer in collaboration with Sunil Hingorani here. We hope to actually have that in the clinic within 1 to 2 years, and we hope to actually have it in ovarian cancer in about the same time frame. So we can, many of these targets, or many of these abnormal pathways that cancer cells hijack, are hijacked uniformly by lots of types of cancer cells. And so if you can have an immune response that targets it, you can actually probably target many different types. So we're trying to understand, one of the things that different cancer types have are different ways of trying to protect itself. So we're trying to understand how those tumors, for example, pancreatic cancer and lung cancer, protect themselves so that we can potentially modify the cells so that we can make them even more effective in those settings.
All right, thank you. What do you think are some of the biggest challenges right now to getting immunotherapies like yours to patients? And what solutions are people exploring right now to help overcome those challenges?
Well, the biggest problem, of course, now is just getting this technology perfected and out there. And I think as Carl June talked about when he spoke in a webinar in this series, this is a project that's really been pursued entirely in academic laboratories. It was a technology that everybody thought was just not viable for the pharmaceutical industry or the biotechnology industry because it was going to require too much individual and personalized manipulation. Well, so as a result, it's taken the kind of funding we've received from the National Cancer Institute and from foundations like the CRI that have allowed a small set of labs actually to really push much of this forward. And because of that now, this technology really is available. And our laboratories are pushing this, and now we are really getting a lot more interest from biotechnology groups and from pharmaceutical groups that are starting to get interested in how we can actually bring this, make this more generally usable and translatable to patients. And I think there's absolutely no question that this is going to be a feasible approach for patients that despite this initial investment in cost in developing it, will actually ultimately probably lower the overall cost of treating cancer patients than we are now with the kinds of therapies we have. Because the real advantage here is that these therapies look like they can be curative. And if a therapy is curative, it saves an enormous amount of money, not just lives, but an enormous amount of money in unnecessary therapies that patients really are having to take now.
Yeah, like at the recent ASCO meeting in Chicago earlier this month, there were several reports of very promising immunotherapies, which for all intents and purposes were giving patients cures. It seems that whereas for a very long time there's been a reluctance on the part of scientists and clinicians to use the C word, the other C word, the cure word, because they weren't seeing these sorts of responses. Now with the advent of immunotherapy, and where you're getting these very durable, long-term time frames of protection of cancer recurrence, they're starting to use that word a lot more often. So I'm really glad to hear that. One of the questions that we also just received was, how long does it take?
No, what is that? I said, and we're really glad to be seeing that, of course. And of course, we are concerned about using the cure word because it takes a long time to be able to say that. But we have every reason from the biology that we understand that is a real potential with these therapies.
Right. It's no longer a dirty four-letter word, right? So one of the questions we have here is, how long does it take to produce these T cells outside of a patient before you can then reach a number that you can inject back into the patients?
Well, that's part of what's become so remarkable, this technology. So when we first started doing this, when we first started treating patients with cells that we expanded in tissue culture in the 1990s, it took us about 4 months. And then we thought we had made this enormous advance when we now can do that in about 12 weeks. But now that we're putting genes into T cells rather than having to isolate the cell from each patient, we can actually have cells available for therapy within 1 to 2 weeks, and we even have some technologies that are making it look like we may be able to have cells in less than a week. So this is, there's no question this technology is moving so rapidly that having this therapy available in a very quick time in a patient is not going to be an obstacle at all. It will almost unquestionably be able to be done in less than a week within the very near future.
That's very good news. We're almost out of time, but I do have one more question. In your presentation, you mentioned that prior to giving your immunotherapy, you deplete the patient's immune system with chemotherapy or possibly radiation. I don't recall if you mentioned radiation. That seems counterintuitive when you're giving someone a treatment that requires their immune system to work. So can you explain a little more about why you deplete a patient's immune system before you administer your particular type of immunotherapy?
I will, and that's a really good question because it gets us to a technology we're working on. So what happens is that when, for example, when people get many viral infections, sometimes your lymphocyte count gets transiently depleted. Or when you get drugs that knock down your white count, your lymphocyte count, you lose lymphocytes. But the body understands how many lymphocytes it's supposed to have, and so it secretes these cytokines, or basically they're hormones that drive the immune system to make more lymphocytes, much like when you generate new skin to replace skin that's lost when you cut yourself. So it restores the lymphocyte counts back to normal. And so the purpose of doing this lymphodepletion with chemotherapy or radiation to knock down the total number of lymphocytes in the body is to essentially create space to make the body think it needs to drive lymphocyte counts under it up. And so what you do is if you give lymphocytes at that time, those cells will be able to harness those hormones and expand and fill that empty space. Now we've learned we're learning very clearly what those factors are. And we actually already have technologies, and we've been working with Stan Riddell's group and Mike Jensen's group on this, that we believe we may be able to do this without having to cause lymphodepletion. We may be able to preferentially let these cells expand without having to do that. And that will be an enormous advantage because you can get rid of this toxicity that you may need to do to get this rapid expansion of cells after you give them. So we think that that's a technology that's been very useful now. We don't think it's actually technology that will become ultimately part of the permanent therapeutic regimen.
Okay, and that's certainly good news because the fewer treatments a patient requires, the less hassle, the less pain, and it's a lot less expensive too, I hope. Because that's certainly an issue that the field is now dealing with as these drugs become approved by the FDA for treatment in patients. So our last question before we go is, what is your particular vision for the role of immunotherapy over the next 10 to 20 years? And how will it really transform cancer treatment?
Well, I think, as we talked at the very beginning, I think the real problem now is that drugs are not adequately selective. And the problem is that tumors are not adequately different, that drugs are always going to have toxicities. And the bigger problem, in addition, is that tumors are going to are able to escape most drugs. That because tumors have unstable genomes, they're proliferating a lot, and as they proliferate, each copy is a little different than the previous copy. That targeting with drugs is always going to be a difficult problem, and it's going to be a problem that's going to have toxicities. We believe that ultimately what we're going to be able to do is harness the immune response so that patients can be treated in ways that will have much more limited toxicities. I'm not going to say it's going to be completely free of toxicity, but it's going to be much more acceptable toxicities, and that more importantly, it's going to be therapies that can make tumors go away and stay away. And so we really see that immunotherapy is going to be a critical part of the armamentarium of cancer drugs. We know now with, for example, monoclonal antibodies, there's many, which is another form of immunotherapy. This is a standard part of many therapies right now. And using the cellular arm of the immune system is going to be the next really potent part of that to really impact how patients' outcomes are changed.
It's wonderful. Thank you so much for joining us today, Dr. Greenberg. Again, Dr. Greenberg is at the Fred Hutchinson Cancer Research Center, the University of Washington in Seattle, Washington, and he's, of course, a very strong player in the field of immunotherapy as well as with the Cancer Research Institute. So we're very grateful that you could join us today and take our questions and share with us some of the exciting developments in cancer immunotherapy. So this is the 5th and final webinar in the month of June as part of Cancer Immunotherapy Awareness Month. We were delighted to be able to present these to you. And you can watch all of the webinars by following the link here, cancerresearch.org/webinars. This webinar today will be up in a few days, but you can certainly view the other 4 now. They're available, and of course free of charge, thanks to our sponsorship from Dendreon. I'd also like to remind everyone that for the rest of the month of June, 1-800-Flowers.com does have a special promotion where 10% of the sale of its Fight with White collection will go to support the Cancer Research Institute. This is a link to that. These are also our Facebook, Twitter, and blog links where if you want to stay up to date on all the latest developments in this very exciting field, these are 3 great ways to get that information to you. Once again, thank you, Dendreon, for your support of this webinar series. And thank all of you for joining us today. You can learn more about Cancer Research Institute's work to advance immunotherapy by going to cancerresearch.org/fightwithwhite. We hope that you'll join us for our next webinar series. We are planning to do some more over the next year, not just in June. So be on the lookout for that. And thank you all.